love to introduce our next two speakers. Jay, if you want to come up too, don't be shy. So our next two speakers are going to be co-presenting our next talk today. Dr. Linlin Guo and Jay Overholser are both from Ohio State University. Linlin is a postdoctoral fellow and senior researcher for Dr. Pravin Kaumaya's lab at the Department of OBGYN at Ohio State University in Columbus, Ohio. His research background includes postdoc appointments at the University of Cincinnati and the University of Louisville for translational medicine of cardiac diseases research. Linlin has served as an editor and on the editorial boards for several peer-reviewed journals. Since 2018, Linlin has also followed Dr. Kaumaya's lab on focusing on cancer vaccines and COVID vaccines, vaccine development, and into clinical trials. For Jay Overholser, he's the lab manager also in Dr. Kaumaya's lab at Ohio State University. He graduated from Ball State University with 2 BA degrees in biology and political science. Jay has over 30 years experience in the lab, working at the University of UT Southwestern Medical Center, also worked for ImClone Incorporated, Memorial Sloan Kettering Cancer Center, and the Ohio State University. So Jay and Linlin, if I'd like to bring you guys up, please.
Thank you for Luminex's invitation. Since we talk about the lottery, right? Nobody won the lottery last time, so as I know, the lottery will go up next time. Hope you guys, somebody will have a lucky can.
See you next year, Linlin.
Yeah, okay. Yeah. Now today I will talk about something about the translation for the B-cell epitope, the peptide-based vaccine. We will translate our vaccine to clinical trials for the immunotherapy. Here, mainly I will talk about our latest vaccine, that's the PD-L1. Glad you just cut off your database searching before October. This one paper just published last month. If you cut off on last month, I maybe have no chance to talk about here. Thank you. As we know, cancer is a kind of deadly disease worldwide. Every year, only in the US, there are over millions of people has been diagnosed, and also over almost 20% of them will be dead. So for the cancer therapy, there are lots of approaches. For the classical one, we normally will be use the surgery or use the chemotherapy, but that's kind of thing maybe when we easily to diagnose them. But the cancer may be caused by the gene mutation or some of the microenvironment reprogramming. That's a condition. It's easy to diagnose the cancer at the early stage. In that kind, we can modify the cancer environment to give them early treatment, for example, the gene editing, or even use the vaccine to modify the cancer environment to give the patient a good outcome. As we know, in the 2018, there's the hallmarks for the cancer immunotherapy. 2 of the top professors and scientists won a Nobel Prize for their discovery of the PD-1 and the CTLA-4 immunotherapy to give very successful on the clinical cell therapy for the patients. Lots of patients got benefit from their discovery. But of course, only a certain percent of patients got a positive response. Most of them have no response or even have a serious side effect. So in this case, we are eager to develop some more advanced strategies and methods to give patients more hope and extend their life. Here, we want to tell something about our lab to develop a B-cell vaccine. There are some more advantages for this approach because we use the B-cell epitope linked with the T-cell epitope. In this case, we can boost the patient's native immune system to use the B cell to generate the antibodies, that's monoclonal antibodies, to against the abnormal cell, that's cancer. And also at the same time, they can activate the T cells to help the B cell against the cancer. This image is the general mechanism how our B cell epitope peptide vaccine works. At first, we generate our peptide vaccine by link the peptides with the T cell here. It's a T cell epitope with linked with the B cell epitope. When we inject in the human, they can both active the T cell and also the B cell. Then after they generate the polyclonal antibodies, the antibodies will against the cancer cells. This is our purpose, to use the patient itself immune system against the cancer themselves. This paper was published early of this year and the 2020. This is our PD-1 vaccine. In here, we developed the PD-1 vaccine in combination with our HER2 vaccine here, this one. This vaccine already been approved by the FDA for clinical trial both in Australia and the US. As we report from our founder from Australian imaging company, one of the patients received the PD-1 vaccine, got a very good response, almost have no tumor now. So hopefully, thank you. Hopefully we will recruit more patients to get benefits from our PD-1 vaccine. This is our latest paper published last month. This is my main topic. We'll introduce our PD-L1 vaccine, how we developed this vaccine, how effective our vaccine works for the preclinical trial. Here, first we generate 4 epitopes from the PD-L1 peptide. We link the peptide with the T cell epitope, MVF or TT3, with the linker. After we inject our peptide into the animals or the future human patients, both of the peptides, both sides, will activate the T cell and the B cells. In this experiment project, we use 1, 2, 3, 4, at least 7 animal models. That's over 300 animals experiment to verify our vaccines. We primarily immunize the animal at 6 to 8 weeks old mice. After 3 weeks, we will give them the first boost and the second boost at more than 3 weeks later. After the last immunization, we will challenge the mice with the designed tumor cells. And then we will monitor the tumor cell growth during the period. For the control group, we will treat the mice with PBS or the golden standard monoclonal antibody. Here, this one, we will treat the mice with our 10-IFG92 monoclonal antibody. This is very widely used for other journals. From here, we can see after we immunize the mice, all the mice have generate high titers antibodies, and also the antibody have good immune activity with the recombinant protein. For further validate which epitope works better, we use the CT26 cells model to see which epitope will give the cells proliferation inhibition. Here we can see the first one, PD-L136, and the PD-L1 have a good response to inhibit the cell growth. Here, this is our further study. We treat on the mice model. We use the CT26 tumor model to test all the 4 epitopes. As we can see clearly here, this is the PBS control. This is the monoclonal antibody control. We can see the immunized mice have the similar and comparable tumor inhibition ability with the control group, and with very less tumor burden than the PBS group. For further validate how about our vaccine works on other tumor models, we choose the mice 41, that's the TNBC breast cancer models, and also this one is the normal breast cancer models. From these two breast cancer models, we can got the similar results that our vaccine works better or comparable than the monoclonal antibody group to inhibit the tumor growth and especially extend the survival rate. We can see the survival rate normally extend at least double the survival rate than the PBS group. Because our PD-L1 vaccine was linked with the two T-cell epitopes, one is the MVF, one is the TT3. For the MVF1, that only works on the biopsy mice because they have the different MHC expression. But here, we first will test the TT3 T-cell epitope on the biopsy mice because this one will work on the black 6 mice as well. We verify this on the biopsy mice. We got the very similar result, and we can see that the similar tumor models, CT26, D2F2, and also the 41, the vaccine works super great and extend the mice survival rate. And now we will further this study on the black 6 mice. We use the black 6 specific tumor models that's on the MC38, and we also overexpress the MC38 with the HER2 on the tumor cell, and also we use the B16F10 melanoma. As expected, we can see that after we immunize the mice got good response on immunogenicity, and also the tumor has been inhibited, whether we immunize the mice or from the monoclonal treatment group. But the thing is, you can see here, this is similar as our clinical trials. Only some of the mice treated with the monoclonal antibody have a good response. Some of them still have very big tumor. So that's why we want to boost the native immune system to let the mice and even the patient against the cancer by themselves instead of the drug itself. Now, here I want to talk about the combination treatment, because the combination treatment is the treat of the cancer therapy. As we know, the cancer avoid be attacked by the immune system by various of strategies. If you block a PD-L1, they might use the CTLA-4. If you block the CTLA-4, they maybe use the HER2. So lots of things are unexpected. So we will try our best to block as much as signaling as possible. Of course, we will know which gene is especially high expressed in a specific cancer. That's why we need pre-screening and use the personnel strategies to kill the cancer in the future. Here we want to talk about our strategies against the D2F2E2. That's a breast cancer tumor cell line. We express the human HER2 on the tumor cells. Here we immunize the 4 times, not just because we immunized the mice at the beginning of the pandemic, but then, unfortunately, the SARS attacked us. So the university wanted to shut down the lab, but we told them, oh, we are developing a vaccine. We need to challenge our mice. So we extend one more immunization. We give the mice the 4th immunization and challenge them. Very glad to see the results after we give them one more immunization, but this is not the standard. We normally give them only 3 times. After the tumor challenge, we monitor the tumor growth. This kind of tumor cell line grows a little bit slower than normal. We can also see that after the immunization, both of the HER2 and the PD-L1 had high titer antibodies. And also, this is the polyclonal antibody. So that's why we also tested the subtype of the antibodies. We can see the IgG1 and IgG2 also have a very high percentage. Here we can see, this one, we can show that after the tumor challenge, I bleed the mice at the 8 weeks of the tumor challenge. That's after 2 months. We can see after the 2 months, the sera still have high titers against the PD-L1 and also the HER2. We can see here is the one without tumor and the one with tumor. The mice with tumor normally have a little bit less antibody inside. So the one without tumor, they still can generate very, very high titer antibodies. So that's why I think if we can boost the antibody with high titers and amounts of antibody in the human, we can against the cancer more effectively. Here, after we can see that the tumor here is very impressive. The combo with the PD-L1 and the combo HER2 itself, and even the combo PD-1, we can see almost no tumor growth, very, very few 1 or 2 mice with small tumor. And also we can see here, the mice after the day 17, that's 9 weeks after the tumor challenge, over 80% of the mice in the combo HER2 group and the triple PD-L1 group almost without tumor and very, very small tumor. All the mice are still survive. So that's another further study I want to show. How about those tumor-free mice or the mice with very small tumor, how they respond to the second time tumor challenge. So after here, I selected the 3 mice from the combo HER2 group and the 3 mice with the PD-1 and the combo HER2 group, and the 5 mice from the PD-L1 combo HER2 group, all these 11 mice without any tumor after the first time tumor challenge. So I bleed them again to test how about the antibody titers in the serum, and then give them the one more boost to determine whether we need to challenge them or not. So we can see here, the mice immunized 4 times and 1 more times boost, they generate very high titer antibodies against the HER2 and the PD-L1. After we challenge, the second time challenge the mice with the D2F2E2 mammary tumor, that was the 91 days after the first time challenge. And here we can see the mice with the combo HER2 PD-1 and the combo HER2 PD-L1. The mice response is super good. Even the second time tumor challenge, most of the mice, at least 1 or 2 mice in of them, that's about 60% and 30% of mice still without tumor. So we can, from this data, we can see that single vaccine works good, comparable or better than the monoclonal antibody treatment group. But the triple combination therapy is the best option, no matter from our preclinical test or even in the future clinical trial. Now, what's the mechanism our PD-L1 vaccine or our vaccine works, how they kill the cancers? So the thing is, of course, they inhibit the tumor growth. Why? So we tested our PD-L1 vaccine, generates the antibody. We use the human BT474 tumor cell line to test the tumor growth. Here we can see that at the high dose, 0.5 microgram ml, all of our vaccine was against tumor growth. And of course, this atezolizumab was our positive control. And here we also test the apoptosis. Apoptosis can lead to the cell death. That's one of the common mechanism against the cancer. But of course, this one we need to avoid for our normal cell. We need our normal cell to survive, and especially the T cell against the cancer as well. So we can see here the vaccine and the antibody and also the atezolizumab, they can lead high percentage of apoptosis of tumor cells. And also ADCC is a very important mechanism. That's the antibody binding at the tumor cells and then recruit the T cells to the tumor cells against the cancer. So we can see here, we use the atezolizumab and atezolizumab as the positive control and negative control. This is the commercial one. One of them we know is can lead to the ADCC. One already been modified, have no ADCC. But our antibody we can see here, at the high dose, 3 of them can lead to the ADCC, but the low, medium dose, we can see only the 36 and the 130 can lead to the ADCC. But of course, the monoclonal antibody group, commercial one, is the highest one. That's the monoclonal antibody. Ours are polyclonal antibody. Even that's not exactly comparable, but we can see here our antibody works against the cancer with a similar mechanism. Here is the best advantage we can see here. We don't want to see any systematic cytokine storm has been released after we immunize the patient against the cancer during the therapeutic period. So we use the panel from the Luminex panel to test the time period of the cytokine release. We tested the serum at the point of the tumor challenge and also 2 weeks after the tumor challenge to test which kind of cytokines have been boosted or they systematically boost. Glad here we can see there are no significant cytokines has been overexpressed during this period. Because this is a kind of, seems like a negative result. The only thing we want to show them, the vaccine was safe, no toxicity has been induced to the animal. I think this might be the best advantage for the clinical trials because the patients receive the monoclonal antibody treatment, they don't want to see any adverse event. So use a single dose of serum, you can test over 20 around cytokines release. So that's very, very, very effective. So I think this kind of method is very important for clinical trial use. For the lab, of course, we test this cytokine release in this experiment and also in our previous PD-1 vaccine development. And also we still have our COVID vaccine ongoing. So all of this, we use the cytokines panel to test our vaccine were safe, make sure they are effective and safe to the patient. Of course, previously we want to save to the animals. Here, like I said, we need to block several kinds of signaling pathways to inhibit the tumor growth. Because tumors, they are smart. Because they have various ways to avoid attack by the immune system. So we need to try to block them as much as possible. We use the HER2 here and the PD-1 or PD-L1 to block their signaling transition and to inhibit them at the beginning. To further verify the mechanism of the PD-L1 vaccine works, we tested the cell-based PD-1, PD-L1 blockade assay. Here, I want to show here. The thing is the basic mechanism here is the PD-1, PD-L1 blockade. If you blockade the signaling here, the PD-L1, PD-L1, if you block this interaction, the signaling will be induced. If they interact with each other, there are no signals at all. So you remember this, it's easy to understand how this works. We first will transfect the cells, grow the PD-L1, and then we use the antibody, PD-L1 antibody, incubate with the cells. The specific antibody will binding at the PD-L1 on the cell surface. And then we add the effective cells to incubate with the previously added the monoclonal antibody or with our polyclonal antibody. If the antibody binding with the PD-L1, they will block the PD-1, PD-L1 interaction. So the signaling will be induced. As we can see here, the monoclonal antibody atezolizumab was very effective to induce the signaling, and our PD-L136 and the 130 can also effectively blockade the PD-1, PD-L1 interaction to induce the signaling. But you can see here the data in some degree varies a lot, so it's not very easy to determine. It's really, really how effective it is. So the next point, we want to find an effective way to find out how effective the PD-1, PD-L1 interaction has been blocked. Here, we normally use the ELISA-based PD-1, PD-L1 blockade assay. From here, we can see that we increased our dose to 2 microgram per ml and 0.5 and also 0.125. And we can see here still, the atezolizumab is also the very effective one. That's no doubt monoclonal antibody. But our polyclonal antibody was also very effective. We can see here the 51 and the 130 and the 95 and also 30, they are very effectively to block the PD-1, PD-L1 interaction. But here, I want to show you what's the mechanism, how this assay works, and also why we need some further assay to verify our experiment. Here, there are two kind of ways to detect the PD-1, PD-L1 interaction. The first is we coat the plate with the recombinant human PD-1 on the plate. And then after the overnight coating, the PD-1 was coated on the plate and the free one will be washed. Because the PD-1 was on the plate, we need to detect the PD-L1 interact with the PD-1. So outside of the well, we incubate the anti-PD-L1 antibody together with the built-in labeled human recombinant PD-L1. If these two will interact and bond together, we can find here the antibody bound with the human recombinant PD-L1, and then this one is no binding. Because this is a computation, maybe we need to increase the time to make them binding if we want, but we will cover the time, cut the time at 1 hour to check the computation results. Here, and then we add the mixture to the wells. If the binding site has been blocked, this one will not bond with the PD-1. But the free one will bond with the PD-1. So we can detect these signals in the later stage. Here is another kind of similar mechanism, but we will switch to coat the PD-L1 on the plate. Here we can see if the PD-L1 was coating on the plate, we will not incubate the PD-L1 with the protein outside. We need to put the diluted serum in the well to compete with the binding site. So after the anti-PD-L1 antibody bound with the PD-1, with the PDL1, this one will not interact with the PD1 built-in labeled recombinant protein. So after wash, this one's still here. But there are still a lot of unspecific binding sites living there. So in this case, some of the recombinant PD1 built-in might be still bound somewhere else or will not be bound there because it has been interrupted by the unspecific binding. So in this way, these two different ways to detect the PD1, PDL1 interaction give us a different result. The one I show here was the first method we detect. We call it the indirect competition ELISA assay, PD-L1 blockade. This one, we use the indirect because we washed this free one away. We left the binding one on the plate, and then this one, we keep this here. So the result completely different. But this is the way we test on the ELISA. Another kind of important thing is, sometimes we also test currently we screen the tumor. We use the cell-free tumor cells. Those cell-free tumor cells, they are released from the primary tumor cells and they're circulating in the serum. Of course, we can use that kind of thing to modify the tumor-free, the cell-free tumor cells in the blood to test their interaction. So in order to further verify our blockade assay, we developed the microbead to use the Luminex, the microbead sphere to further verify the PD-L1, PD-1 interaction. So the next step, I will introduce my colleague, Jay over here, sir, to give you some more detailed method and the procedure about our PD-L1, PD-1 blockade assay, how we prepare the beads, and also how we test them, why we tested this to here. Thank you.